is it? From cell structure, we started some of the questions we have solved, and today again we will continue from that. All right. Um, this is say here question number. We did, I think, in the previous class we did up to serial number 16, is it? We did up to serial number 16. So today, next one, number 17, right? Number 17. What is true about mesosome? Mesosome. I think in the previous class also I have explained something that mesosome in question number 13 say a sliced differentiated form of cell membrane of prokaryote is the mesosome, this structure, is it? So here regarding mesosome, another question, what is true about mesosome? It helps in the cell wall formation, it helps in cellular respiration, it helps in DNA replication and all of the above. So this is true that all of the above will be. It is for the cellular respiration also. It take part and it also help in DNA replication because during the DNA replication, DNA is attached to this mesosome. Okay, and then when the DNA replicate, the cells divide in such a way that one DNA goes to one cell and another DNA copies to another cell. So here it is that all of the above. Here it is one thing that select the mismatch. Gas vacuoles, green bacterial cells, large central vacuole animal cells, protist, eukaryote, methanogen, prokaryote. Which one is wrong?
Number two.
Number two, large central vacuoles. In animal cells vacuoles are very small. Vacuoles are very small or it is absent also. But in case of plant, it is true for the large central vacuole is true for, this one is true for plant cell, not true for animal cell. For plant, it is true. Okay. And this one, see gas vacuoles, green bacteria cell. These things, there is a gas formation is there that particularly that cyanobacteria, you know, cyanobacteria, that blue-green algae, in cyanobacteria, these are all those which photosynthesize. Okay, they have this gas vacuoles, and particularly in case of cyanobacteria, as when they carry out photosynthesis, this cyanobacteria, they produce oxygen. Okay, and due to the production of oxygen, they become light and they float. Of course, here this is a gas vacuole where gas is stored. This one in the green bacteria cell. Of course, the green bacteria cell, bacterial cell, they do not produce oxygen. Remember, those bacteria, bacterial cell, green photosynthetic bacteria, no? Photosynthetic bacteria, green purple bacteria, they do not produce oxygen, of course, but they have gas vacuoles. Protist, protist are the kingdom Protista, member of kingdom Protista. They are eukaryotic organism. And methanogens, methanogens are methane producing bacteria. Is this? They are all prokaryote. Methanogens are also used for those gobar gas plants and other things because they produce methane. No, during the decomposition of logs, plants, etc. Sometimes methane is produced. So methanogen they are found in deep area where there is a decomposition in coal mines, etc. Also, these methanogens are found. Next question. So our here is the this one question option 2 correct. Correct means it is a wrong statement mismatch. 19. Select the incorrect statement about prokaryotic ribosome. Sorry, here this incorrect, this one is incorrect. It should be incorrect. Okay. This should be incorrect one word. Incorrect statement about prokaryotic ribosome. One is 50s and 30s subunit unite to form 70s ribosome. Polysome consists of many ribosomes attached to tRNA. Ribosome is the site of protein synthesis. Polysomes indicate the synthesis of identical polypeptide in multiple copies. See one thing, in a ribosome, ribosomes are important cell organelles without limiting membrane. Okay, ribosome don't have, these ribosomes are made of rRNA and protein. All right, ribonucleoprotein structure, more or less some granular structure, and these ribosomes are made of two subunit. These two subunits, one is a large and one a small subunit. Here, this ribosome, their size is different in prokaryote and eukaryote. In prokaryote, it is 70s, and eukaryote, it is 80s. S is the sedimentation coefficient. Okay, so this is a physical property. So this is 80s, this 70s. So here, this 80s and 70s, they are made of 50s of large subunit and 30s of a small subunit. This is true. Actually, if you see this 50 plus 30 means it should be 80, but no. Sedimentation coefficient S is a value which is determined. This is a physical property like density, mass, no, a specific gravity, those type of things. And this is not additive, that two things having 40 and 60 you add, it will be 100. No, they are not additive. This large subunit, when you determine its S value, it will give 50S. And its a small subunit, its S value, when determined, then it comes as a 30S. But when these two subunits join together to make a complete ribosomal subunit, and then its S value is determined, that S value will come to 70. Okay, so 50-30 it is. So prokaryotes have ribosome 70S type, and their subunit are 30S and 50S. Eukaryote have the ribosome of 80S type, and their larger subunit is 60S, and a smaller subunit is 40S. Okay, this 60 plus 40 is not 100S. They make 80S. This is in eukaryote. This ribosome is found free in the cytoplasm. Ribosome is found in the prokaryote. In the eukaryote, ribosome is found within the cell organelles. Like ribosome is found within, that inside the mitochondria, and also inside the chloroplast, that is in plastid. So ribosomes are there, and in chloroplast and mitochondria the ribosomes are not 80S type, they are 70S or less than 80S. Okay, this polysome here written attached to tRNA. No, during the ribosomes are the site where the protein is synthesized, and during the protein synthesis, that is the polypeptide, it is synthesized as polypeptide. Okay, polypeptide is, this is chain of amino acid. When the different amino acids are joined together by peptide bond, amino acids are joined together by peptide bond and form the structure, the chain of amino acid, that is known as polypeptide. Polypeptide make the protein. Okay, so when the ribosomes are active in this polypeptide synthesis, at that time the ribosome is attached to mRNA. Okay, ribosome attached to mRNA, because in the mRNA, M stand for what? Do you know? M stand for messenger.
Messenger.
Yes, so this RNA is a messenger RNA. It carries the message from the gene, from DNA. All right. And according to the message, the amino acids are synthesized, arranged. Who read this message? The ribosome read the message present in the mRNA. So one ribosome will move along the mRNA and read the message, and accordingly amino acids are joined. So when one ribosome will move from the initial point to the end, then one RNA, one polypeptide will be synthesized. All right, one polypeptide will synthesize. One ribosome, it will help in synthesis of one polypeptide. But what happened that during this protein synthesis, we can see that with the 2-1 mRNA, many ribosomes are attached. Say here 3 ribosomes attached and they are moving forward. So this one will synthesize one polypeptide, this one will synthesize another polypeptide, this one will synthesize third one. Okay, but as it is the same mRNA, so same message is there. So the polypeptide that will be synthesized by this ribosome will be same. So multiple copy of polypeptide will be synthesized. This indicate that there is a synthesis of multiple copy of polypeptide. This is called polyribosome or polysome. Okay. So here that this one is this to the RNA, tRNA, this is wrong. Polysome consists of many ribosomes attached to tRNA. This is wrong. So incorrect statement is this one, number 2. A ribosome is the site of protein synthesis. That's true. Okay. Polysome indicate what? Synthesis of identical polypeptide in multiple copies. Yes, this is synthesized in multiple. As I told here that they are synthesizing each ribosome as it will move from one end to the other end will synthesize one polypeptide, and this all these polypeptides are from the same mRNA, so these polypeptides are identical. Okay, so these things are all correct. Only the choose the incorrect pair. Now question number 20. The best material to study cell membrane structure. Here this side matching which is correct or incorrect. RBC of human. Arrangement of lipid in cell membrane, a bilayer. Abundant lipid in cell membrane is the phospholipid. Cell membrane composition, carbohydrate, lipid only. Cell membrane composition, carbohydrate, lipid only. Say when the plasma membrane structure and its properties were studied, it was membrane from the RBC, red blood corpuscles. Okay, the plasma membrane of RBC were isolated and they were studied. So this matching is very much correct. Best material to study cell membrane structure is RBC of human. From here, plasma membrane were taken. Arrangement of lipid in cell membrane. The lipid that they form two layer lipid molecules. Okay, lipid molecules are arranged two layers, not one layer. So we call lipid bilayer. This is lipid bilayer. The plasma membrane is made of lipid, and along with that, the important other component is the protein. That is why we call sometime that plasma membrane is a lipoproteinous. Lipoprotein structure, that is lipid and protein within this, but lipid form the continuous layer, and within this the plasma membrane, the proteins are embedded. So this one also is true. Abundant lipid in cell membrane. The lipid type of the lipid which is found maximum amount. There are lipids are also of many different types. The one which is present frequently or in higher quantity, that lipid is phospholipid. That is the phosphate containing lipid molecule, phospholipid. Cell membrane composition, here it is carbohydrate, lipid only. No. Main is lipid and protein. Okay. Lipid and protein is the main, and carbohydrate is very less. It may be 5% or less. All right. Carbohydrate. So this one is a incorrect match. It is lipid, protein and carbohydrate, but main is lipid. So incorrect pair is number 4. I will show you that the lipid structure, lipid molecules. See, this is the structure of plasma membrane, the fluid mosaic model. Can you see this fluid mosaic model of plasma membrane? This one was given by that this is the most accepted method, or this theory or hypothesis. It was given by Singer and Nicholson. Okay, they gave this model, and this is called fluid mosaic model, a structural model. And in this, you can see this blue portion, white and blue portion, these are the lipid molecules. So there is a continuous mass of lipid molecule, and within this lipid molecule there are this red color things you can see. These are the protein. Proteins are embedded either partially, say like this one is partially embedded, but this protein molecule is embedded completely. Okay, this this is called the integral protein, and the proteins have come out on the surface on both the side. Okay, this is here. Can you see that with the protein, long thread of protein, you can see that bead-like thread are there. This is the carbohydrate. The small molecule of carbohydrates are there. A small chain of carbohydrates are there. These are this is present. Carbohydrate is present only on the outer face of the plasma membrane or biomembrane, not on the inner face. Say this one is towards inside the cell. So this side the carbohydrate is not there. Carbohydrate is present on the outer surface of the plasma membrane. Okay, this one. So this is semi-fluid, and this molecule can move. Okay, I will show you another diagram which will be more clear. Yes, this one you see. This one is more clear. Okay, here, here. This round ball-like structure. This ball-like structures are mm, this ball-like structure. These are the this lipid molecule. This lipid molecule has two portion. Okay, two part. One is head. This is polar. Okay, and there is tail. This one chain of hydrocarbon. This is the tail. The tail is non-polar, and this one is hydrophobic. Hydrophobic. Hydrophobic tail and hydrophilic head. This head, this is polar head, it is hydrophilic. Okay, so it has affinity for the water. And this tail, it is a non-polar, it is hydrophobic and called tail. There are two tails are there. So these lipid molecules are arranged in the plasma membrane in such a way in lipid bilayer that their tails are facing inward away from water because this is hydrophobic, so it has to keep away from water. So tails are on the inner side and heads are exposed. The polar heads are exposed on the outside. So it is keeping, protecting the tail portion, hydrophobic tails. They are arranged and this lipid molecule forming the continuous layer, lipid bilayer, and within this protein molecules are present here and there. See, this is a protein molecule, this is a protein molecule, this one is protein, this one is protein, showing that this protein may be from one surface to the other surface, it is exposed, totally embedded and extending from one surface to other surface. This also, this one you can see that protein. So there are two types of protein. One is called integral protein. Okay. Integral protein, they are embedded partially or fully. Some proteins, they are present only on the surface of the plasma membrane. Okay. Hello. Hey, who has drawn the line? Yeah. Please remove that. And these are also protein. And to the protein, this is carbohydrate. The small sugar molecules are there. Carbohydrate. Carbohydrate portion is very less. So this is the structure. This is called the fluid mosaic model given by Singer and Nicholson. Model given by Singer and Nicholson. 1972. Okay. So this is you have copied now. Okay, now come to the next question. See here the question that what I have explained all. The lipids are arranged within the membrane with polar head towards inside and hydrophobic tail towards outside. No, this is not correct, is it? Both heads and tail towards outside. How can it be head, tail, both outside? Heads towards outside and tail toward inside. This is the lipids are arranged within the heads towards outside and tail towards inside. Both heads and tail. No. So our answer is 3. Question number 22. The lipid component of the cell membrane consists of lipolipid, phosphoglycerides, hydrophobic lipid, none of these. This one is the phosphoglyceride. Okay. Phospholipid is mainly maximum, no? Phospholipid, that is the lipid is made of glycerol and okay, it is phosphoglyceride. Number 23. Why tails of lipid in the membrane are towards inner part? Why it is towards inner part? Tails, tails of the lipid, because the tails are hydrophobic. So which one will be correct? The non-polar hydrophobic hydrocarbon tails of lipid being on inner side ensure their protection from aqueous environment. Is that? The non-polar hydro or hydrophobic hydrocarbon tail of lipid, that is the tails of lipid being inner side, what happened? It ensure the protection from, because it is hydrophobic. Next, according to modern concept, cell membrane is, this is called quasi-fluid structure, plasma membrane or cell membrane, or we call biomembrane. Why we call biomembrane? Because say this we call biomembrane because not only the cell membrane, the membrane of the nucleus, membrane of endoplasmic reticulum, membrane of the mitochondria, membrane of this Golgi complex, the membrane of the chloroplast, all have the basic structure, that is lipid bilayer. All right, lipid bilayer with protein. So that's why I call the biomembrane also. So according to modern concept, this cell membrane is quasi-fluid structure. Okay, this is a quasi-fluid, means it is a fluid, but not a fluid like a water that it will flow, because this has to give a definite shape to the cell organelles, to the cells. Okay, it is a limiting membrane, so it cannot be total completely fluid structure, and it cannot be very solid, hard, solid also. Okay, so it should be in between, semi solid type of a structure. And here, so it is called quasi-fluid, and in that what happens, those molecule of the that lipid molecule, they can make movement, lateral movement also, and there is soft. Sometime if a cell is pressed from here, so it will fold or it will bend. The flexibility remain, but it will not get torn. Okay, or if it will get torn also, then the say if this plasma membrane is torn here, injured here, then what will happen? The lipid molecule from the side will move laterally, and they will fill up the gap. So this is the fluid, that quasi-fluid structure. Next, the fluid mosaic model explain what? Only a structural aspect of cell membrane, only functional aspect of cell membrane, Yeah, structural also, functional also, and structural and functional aspect of cell membrane. So both structural and functional aspect of membrane. Fluid nature of membrane is able to explain what? Due to this fluid nature, cell growth is possible, cell division is possible, because you remember that during the initially cell is small, then it will grow, so the same cell will grow like this, so new membrane is produced and added. So during the cell growth it is possible. Cell division, when the cell division takes place, a nucleus has divided. After that, cell will, there is that invagination, and ultimately it grow, and ultimately the two cells separate. So this cell division also it is. Then intercellular junction, two cells are meeting, and sometimes they join, they exchange. So the cell junction is possible by this secretion from the cell. Okay, some of the material synthesized, they have to come out, is it? So how to come out? Or sometime they will engulf some food, something they have to take inside, how it will move in, all these, that permeation of various substances across membrane, all these are also possible due to this fluid nature. And so correct is all of the above here. Next, choose the incorrect statement about the cell membrane. Generally, a smaller molecule pass easily without energy requirement by passive transport. Through the plasma membrane, a small molecule can go. I think we have in the physiology also, in transport we have learned that a small molecule they can go, but for the larger molecule they need a special help. Okay, so it can pass by passive mode of transport. Water soluble substance pass through it. You have to select the incorrect statement. Water soluble substances pass through it less rapidly than lipid soluble substances. Say, plasma membrane is made of lipid, oil, water, and lipid. This is oil, fat oil. Okay. So water and oil does not mix, no. So that's why water soluble substance cannot move easily across the plasma membrane. But lipid soluble substances can move across the membrane easily. So lipid soluble, neutral solids are those which are not charged. Okay, those which are not charged, those neutral solids, they can move by simple diffusion. They can move. This is, so all of these three are correct. So there is none, no statement is incorrect. None of the above. None of the above is incorrect. Another, within the transport, within the plasma membrane, there are a transport system. which is called the pump, sodium potassium pump. Say this one will be, this positive sign will be here. Okay, sodium potassium pump. This sodium potassium pump is present in the plasma membrane. So this is called sodium, and this required to operate this type of pump require energy expenditure. Okay, so it is a active transport. Passive transport are those where no energy is spent. That is, ATP expenditure is not there. Choose the odd one out with respect to the uh, composition with respect to the composition of plant cell wall. Odd one out. Cellulose, galactan, pectin, protein. Cell wall. Cell wall is, plant cell wall is made of cellulose, is made substance. Beside that, there is hemicellulose is there. Pectic substances are there. Okay, and some protein. Pectic substances are in the middle lamella and also in the main cell wall it is found, but their amount is very less. Main is cellulose. Galactan is odd here. Okay, galactan is odd in this case. Of course, galactan is found. Galactan also is a type of carbohydrate which is found in algae, of course. In some of the algae, they have galactan, cellulose and galactan. They don't have any cellulose. Innermost portion of mature plant cell wall is the cell wall is there is primary cell wall and a secondary cell wall. Okay, when this say this is a cell and around this cell wall, say if around this the cell wall is formed, the cell wall material deposited first initially during its earliest stage, that one is primary. But later on, till this cell almost remain living, they keep on depositing the cell wall material, okay, on the inner side. But it will be always outside the plasma membrane. So the primary cell wall is on the outer side and the secondary cell wall will be on the inner side. So the innermost portion of the mature plant cell wall is what it is? Innermost is secondary cell wall. Which of the following is correct for middle lamella of the eukaryotic cell? Middle lamella, you know, that in between two cells, in between two cells, there is a layer. That layer is called middle lamella. So, what is true about the middle lamella of eukaryotic cell? It is formed as a cell plate during cell cytokinesis. Do you remember that when the cell division takes place, say karyokinesis has taken place. After that, cytokinesis will take place. And in case of plant cell, cytokinesis takes place by cell plate method. That is, cell plate are formed. This is actually a small cell wall material packed granule vesicles are deposited. This forms the cell plate, and the cell plate contain which material? Cell plate contain the middle lamella material, that is pectin. Pectic substances are deposited. So it is deposited in the form of cell plate. It mainly consists of calcium pectate. This is pectin, but it is a salt of calcium. So it is called the calcium pectate. Ca pectate written here, calcium pectate. So this is true. It holds different neighboring cells together. Yeah, middle lamella is the cementing material between the neighboring cells. So all of these are correct. Here one that question regarding this also. One question you can see here. This is the diagram of the cell membrane. No, identify the component labeled A. This one in the given diagram of cell membrane. What is this A? Can you tell? This A is what? This is sugar,
Sorry.
is it? Sugar, that is carbohydrate. So with the diagram also question can be given. One very common question, that is the cell wall and middle lamella are traversed by what? When the two cells are there. Okay. Say here two cells, and this is, say, if I draw there is a line. This line is middle lamella, and this one is the cell. This is the cell wall. Okay. Now this cell wall as well as middle lamella are traversed by in between two cells. Say if I erase here, and then draw a connection that this connection is passing through both cell wall as well as middle lamella. What is that called? These passages, these structures are called plasmodesmata. Okay, this is plasmodesmata. And through this plasmodesmata, neighboring cells are connected. Okay, so today up to here, say there are many questions in the cells, and in the next class also I will all the question. It is not possible. Some question in between important one only in the next class I will do, and you have to practice at home as many you can do. So in the next class, so what I will do, that one or two question for each of the cell organelles or things that we will discuss here, and rest of the question you have to do at home. You practice, and if any problem you can bring those problem to the class and ask me. Okay, so today I will stop it here. Do you have anything to ask, any question? Hello. Okay, then I will
No,
stop
sir.
it here. Right, I'll stop here.
Thank you, sir.